Okay, good afternoon everyone. Welcome to lecture 11 of BMS 300. We're going to do part 2 of the connective tissue and just wrap up with that before we move into the very first organ system, which is the integumentary system, and personally I think it's my favorite organ system, kind of competing with cardiovascular as well. So here's an overview of what we'll be discussing. This is a slide I walked you through yesterday when we talked about the various subdivisions of connective tissue. We saw that there are 3 types of connective tissue. You have connective tissue proper, supportive, and fluid connective tissue. Today we'll be talking only about fluid connective tissue. But as we went through these subdivisions, we see that how one can differ from the other is in terms of the cells which are found in that connective tissue and also the matrix which surrounds those cells, right? So and also depending on the type of matrix which surrounds the cells, it will influence the structure of the connective tissue and that can impact its function. So that's something we'll discuss as well. So the 2 types of fluid connective tissue will include blood and lymph, and the question you should be asking yourself is, what are the cells I would see in blood, and also what surrounds the blood as well. So in terms of fluid connective tissue, we see that the cells are going to circulate in a liquid extracellular matrix, and these cells could include red blood cells, white blood cells, but there is this liquid extracellular matrix for both lymph and blood. And what I want to do now is just briefly tell you how lymph is made from the blood. Right? I'll explain how lymph is made from the blood. So here's a cartoon of the heart, right, showing that it's going to pump this blood which is in blue color. It's indicating the deoxygenated blood. It's sent to the lungs, and in the capillaries of the lungs it's going to pick up oxygen. It will get oxygenated, thereby they're showing it in red color now. So this oxygenated blood comes back to the heart, and from the heart it's going to be pumped to the rest of the body with pretty high pressure. And so because blood is being pumped from the heart with pretty high pressure, we see that the blood pressure is going to cause some of the plasma to leak into the interstitial space. So as the blood is flowing through the vessels, because of the pressure, the fluid component of the blood will move into the interstitial space. The interstitial space is referring to the space which surrounds individual cells and also the space surrounding the blood vessels. So that is the interstitial space. And most of this fluid will be drawn back into the blood vessel because of the osmotic pressure exerted by the proteins in the blood, right? So because of the osmotic pressure exerted by the proteins in the blood, most of this fluid will move back into the blood vessels. But a small amount of this fluid in the interstitial space moves into the lymphatic vessels. And the moment this interstitial fluid moves into the lymphatic vessels, that's called lymph. And then you'll see that this lymph is going to flow through the various lymphatic vessels, and eventually it'll be sent back into the bloodstream. Right? So that's how lymph is going to be produced. Now the type of cells we see in the blood, I already mentioned. You'll find red blood cells and white blood cells, and in the lymph you're going to find lymphocytes, which are a type of white blood cells. Right? And we'll look at the details of how the blood accomplishes its function, and also what lymph does when we get to blood and the lymphatic system. So this will be several weeks from now. So let's now talk about the functions of the different types of connective tissue, now that I've explained what are the cells which make them up and also how their matrix is made as well. So in terms of the connective tissue proper, we find that the main function is to connect and protect. An example of connections would be what dense regular connective tissue does, right? So dense regular connective tissue, an example of that is tendons. Tendon will link bones to muscle, and so that's an example of how there's a connection between the skeletal system and the muscular system. In terms of protection, the examples I would point out are fat or adipose tissue and areolar connective tissue. So the fat which surrounds the body can help cushion against physical impact. I also mentioned that areolar connective tissue has got the the ground substance, which is this viscous jelly-like material which can absorb shock as well, right? So we see that both areolar connective tissue and fat are examples of how they offer protection. And finally, we see that the connective tissue proper can also help to store energy reserves. So wherever fat is, we see that it can help store extra energy in the form of triglycerides. So with that, we move on to supporting connective tissues, and we find that cartilage and bone will both offer structural strength, and also it can offer the framework of the body. Without the bones and the supporting cartilage, we'll be like jellyfish. We can enter through any small opening, right? But the structural framework of the body is possible because of the supportive connective tissue. Moving on to fluid connective tissue, we see that the main function of blood and lymph could be help in the transport of dissolved materials. Examples of dissolved materials would include nutrients taken from one part of the body to the other. Also, it could be gases which are transported, and oxygen is necessary for ATP synthesis, you know that, and also carbon dioxide is transported in the blood. And finally, wastes which are produced by the cells can also be transported in the blood as well. We also find that there can be protection from pathogens, which is possible due to this fluid connective tissue. I mentioned that blood contains white blood cells, and also the lymph has these lymphocytes which play a role in the immune function, and that can offer protection against pathogens. Are there any questions on this at all? Okay, if there are no questions, I want to mention one more thing in terms of supportive connective tissue. The synthesis of the blood cells will occur in the bone marrow, right? And that's something we'll consider in greater detail when we get to the skeletal system. So with that, let me just summarize what we've been discussing from yesterday. I told you how the classification of the connective tissue is going to be based on the types of cells which are found in the connective tissue, and also the matrix which surrounds those cells. And based on the type of matrix and the structure it forms, we find that the function of the connective tissue can be a number of things, such as protection, support, energy reserve, tensile strength, offering immunity, transport, structural flexibility, and so on. So here I'm going to try to summarize whatever we discussed from yesterday in one comprehensive slide, right? And I would highly recommend that you try to practice drawing this out a few times, and it should take you around 3 minutes if you try to draw it out from start to finish, and try to do that a few times, and you should have this down pat. So I've pointed out all of the cells here in blue color, and the matrix surrounding the cells will be shown in green, and the components which make up the matrix, namely if there's ground substance or just specific fibers, that will be shown in green or in red accordingly. So we find that the main types of cells in the areolar connective tissue will be fibroblasts, and there will also be a number of immune cells. Now the reason why you need immune cells in the areolar connective tissue is that it's underlying the epithelia, which is exposed to the external environment. So if there's any sort of damage to the epithelia, you want to have immune cells available, and they are present in the areolar connective tissue. Next, in the adipose tissue, as the name indicates, you will find adipocytes, and in the loose reticular connective tissue, and also the 3 types of dense connective tissue, in all of them, the type of cell you find are fibroblasts. And the reason why you find fibroblasts here is that it is a fibroblast which will produce the respective matrix which surrounds these cells and makes up the connective tissue. As far as cartilage is concerned, all 3 types of cartilage, hyaline, fibrous, and elastic cartilage, all of them will contain chondrocytes as the unique type of cell in the cartilage. And as far as bones are concerned, we'll be discussing about 4 types of cells, but the major kind of cell we'll consider is osteocytes found in bones. And then in the blood, you'll have white blood cells and red blood cells. And in lymph, you will have the lymphocytes, which are a type of white blood cells. So that is the summary of the various cells you will find in the different kinds of connective tissue. So now we're going to look at what makes up the matrix which surrounds those cells. So in areolar connective tissue, you'll have the ground substance, which is the made up of proteoglycans interacting with water, and then you also find collagen, elastic, and reticular fibers as well. And an example of where you find areolar connective tissue is going to be deep to epithelia. And later in today's lecture, we'll see how areolar connective tissue will be supporting the epithelia seen in the skin. Moving on to the adipose tissue, there is little matrix found there, and so this is the only connective tissue where we won't talk about the matrix surrounding the cells, and you'll find adipocytes generally subcutaneously, but then if that subcutaneous storage is exceeded, it can start being stored in other parts of the body as well. As far as the loose connective reticular connective tissue is concerned, you will have reticular fibers which form this network-like appearance, and this network-like appearance will help to support very delicate organs. Examples of this would include liver and also spleen, and even other lymphatic tissues as well in the lymphatic system. So now moving on to dense connective tissue, we find that in the first two, regular and irregular dense connective tissue, the type of fibers you see are collagen fibers. And the collagen fibers are arranged in a parallel manner, and that will allow resistance to stretching in one specific direction. And so where you find dense regular connective tissue is in tendons, which are pulled in one direction by a contracting muscle. So in the direction in which the tendon is being pulled by the muscle, the parallel arrangement will offer resistance to stretching in that direction. In contrast, we find that the collagen fibers in the dense irregular connective tissue, they're going to be arranged in a random pattern, thereby it offers resistance to stretching in various directions because the collagen fibers are in a random pattern. An example of where you find this will be in the dermis, specifically in the reticular layer of the dermis. What exactly this is, we'll get to it next week. And then in the dense elastic connective tissue, you will have an abundance of elastic fibers, and these elastic fibers will allow the respective organs to stretch and return to their original shape. And one example of where you will have this type of stretching is in blood vessels. As blood is pumped through the vessels with high pressure, it will stretch. As the blood goes past that specific area, it will shrink back to its original shape. In hyaline cartilage, we find that you will have both proteoglycans and collagen fibers which will combine to give it this hazy glass-like appearance. Just proteoglycans interacting with water will give you this clear viscous appearance, but with the collagen fibers, it gives this hazy glass-like appearance. And the hyaline cartilage will be found at the ends of long bones, and it will serve as a template for bone formation in the fetal development. And you will find these collagen fibers also in the fibrous cartilage, and that will help to resist compression. And an example of where you find that is in the vertebral discs. And finally, the elastic fibers will be found in abundance in the elastic cartilage, and that an example of where you find that is the external ear. Moving on to bones, we find that the matrix is made up of both collagen fibers along with this hydroxyapatite crystals, which are a type of calcium phosphate, and so that will make up this matrix which offers mechanical support, and it also is quite strong. In terms of the blood, you'll have blood plasma, which forms the fluid matrix, and lymph is the filtrate of this blood plasma, and that will be the matrix which surrounds the lymphocytes in the lymph. Right, so this slide is very, very important, so please spend some time writing it out. And my advice to the previous section was play your favorite music, right? I know songs typically last 5 minutes, right? By the time an entire song ends, you should be able to draw this out 2 times. Listen to 5 songs, and you've practiced it 10 times, and I promise you there'll be a question on this in the exam. Sounds good? Yes? All right. So let's do a practice question on the connective tissue. Proteoglycans refers to a large class of non-collagenous glycoproteins, proteins covalently linked to polysaccharides, clear matrix surrounding the fibers, or is it large and highly charged polysaccharides? You can turn to your neighbors, TAs, and answer this question. Wait, yes. What is it? Oh, locked? No, I think it's open. People can answer it now. Yeah. Oh, it's on now? Okay. All right, folks, let's bring it back together. Justin, anyone in your group has a response for us? Yes?
Yeah, it's going to be B, proteins covalently linked to polysaccharides.
Okay, proteins covalently linked to polysaccharides. Most of you picked that. That is correct. The strict definition of proteoglycans is referring to proteins which are linked to these polysaccharides. And these polysaccharides are made up of these repeating disaccharide units. And some of you did pick C, which is the clear matrix surrounding the fibers. That clear matrix is the ground substance which is composed of proteoglycans interacting with water, right? But just strictly talking about proteoglycans, it's the proteins which are linked to the polysaccharides. Is that clear, folks? Yes? All right. So with that, we now get to one of my favorite organ systems, and I think maybe the reason is that we see this system every day, we interact with it, we touch it, feel it, and so this may be a reason why it's one of my favorite systems. So let's look at an overview of how we'll be discussing the integumentary system. The question we'll be answering in today's lecture, and also which we'll continue answering next week, is how do the components of the integumentary system aid in its function? How do the components of the integumentary system aid in its function? Now this question has got 2 parts, right? The first part is what are the components of the integumentary system, and the second part is what are the functions of the integumentary system, and we take these 2 parts and combine it to see how the various components of the integumentary system will help it to perform its various functions. So to go about answering that question, we will first consider the 3 layers which you will encounter when you consider the integumentary system. You have an epidermis, below that you have the dermis, and then there's the hypodermis. And in each of these layers, we'll consider what type of cells are going to be encountered. Right? We'll see what are the various cells which are encountered here. And we'll also talk about the associated tissues in these different layers. So in yesterday's lecture, we talked about connective tissue. Before that, we talked about epithelial tissue. And I told you also have muscular nervous tissue. All these 4 kinds of tissues we will encounter in the integumentary system. So pretty much any organ system you will be encountering the various types of tissues. And then there are also these appendages which refer to structures like sweat glands, sebaceous glands, you have hair follicles, all of those are appendages we'll be briefly talking about. We see that even the nails in our hands and feet, they're also examples of appendages, but we won't be getting to that. So let's go ahead and start by looking at some general facts about the integumentary system. So believe it or not, this is the largest organ in the body. It's not the skeletal system, it's the integumentary system, and it makes up around 16% of the overall body weight. So we find that the skin is composed of 2 distinct layers. And those of you who are paying attention to the previous slide, where you may be wondering, didn't you just say 3 layers? Now you're saying 2 layers. All right, bear with me, I'll explain the reason for that discrepancy. Right, so the first layer is the epidermis. This is the most superficial layer. The word epi means above, and then the epidermis will lie above the second layer, which is the dermis, which is the middle layer. Now both of these layers, the epidermis and the dermis, they're going to sit on top of this third layer, which is technically not part of the skin. It's called superficial fascia, and because it is below the dermis, it's called hypodermis. Right, and this is called superficial fascia. So you may be wondering, okay, what is superficial fascia? How many of you have heard the term fascia before? What is the context in which you heard fascia? Myofascial pain. How many have had that term, right? So yeah, so the myofascial pain is just referring to the pain associated with the connective tissue surrounding the muscles. So myofascia means it's the connective tissue surrounding the muscles, and that connective tissue can have nerves which are a part of it, right? And when those nerves are triggered or irritated, that can lead to myofascial pain. Anyway, so this is just called the superficial fascia, meaning this is the superficial layer of connective tissue. And as far as the skin is concerned, this will be the deepest region. And in the upcoming slides, we will start talking about the epidermis, dermis, and the hypodermis in a little more detail. And in today's lecture, we'll only be considering the epidermis, not all of it. We'll finish our discussion on the epidermis when we come back on Monday. So the epidermis is made up of epithelial tissue. You're all aware that epithelial tissue is covering the external surface of our body. And the type of epithelial tissue you find here is stratified squamous epithelia. Specifically, you'll have a layer of keratin on top of these flattened layers of cells, and so it is keratinized stratified squamous epithelia. Now, the epithelia, the epidermis on whole can be divided into 5 layers or 4 layers, depending on where you look. Right? So, depending on which part of the body you consider, there could be either 5 different layers in the epidermis or 4 layers. Now, the 5 layers would be seen in thick skin. Example is the palm of your hands or the soles of your feet. That's thick skin, whereas in the other parts of your body, it's considered thin skin, and you'll have only 4 layers. And this 5th layer is known as stratum lucidum, and we'll consider where it is located in the scope of the epidermis. So this is a picture showing you the 5 layers of the epidermis. We're going to start superficially and see its stratum corneum. So the word stratum means layer. Corneum is referring to some unique property or characteristic of that first layer. Below that you have stratum lucidum, stratum granulosum, stratum spinosum, stratum basale. Each of these second names are highlighting what is unique about that specific layer. Now I will hold you responsible for all of these names and the order in which these layers are found. And so in order to help you remember this, I have this helpful mnemonic, and hopefully it's a fun mnemonic. It says, come, let's go sunbathing, right? And the convenient thing about this mnemonic is that the first letter of each of these words match with the first letter of the second name for each of these layers, right? So C for corneum, L for lucidum, G for granulosum, S for spinosum, and B for basale. Now this mnemonic is kind of ironic because, man, that rhymed. Ironic mnemonic. Anyway, it's ironic because when we come back next week and talk about skin cancers, I'll tell you that exposure to sun in an unprotected manner is one of the reasons for melanoma and for increased risk of skin cancer. Even when we talk about aging, age-related changes to the skin, it's due to exposure to the sun. So it's important to remember that if you are having exposure to sun, just take the adequate protection needed. All right, so with that, let's go ahead and start talking about the various layers of the epidermis. We'll start with stratum basale and we'll move all the way up to stratum corneum. So stratum basale is also known as stratum germinativum, and this is only a single layer of cuboidal mitotic stem cells. Right? These are cells which are rapidly dividing, and they will give rise to keratinocytes. Keratinocytes are the major type of cells you see in the epidermis. So if this is the major kind of cell, then there should be a minor kind of cell, right? So around 10 to 25% of the cells in stratum basale are melanocytes. These are the cells which produce the melanin pigment. How exactly this melanin pigment is going to be transported into the keratinocytes, we'll discuss it next week. There are also some tactile cells. Tactile cells are referring to sensory receptors, and an example, and the only kind of sensory receptor in the epidermis is the Merkel cells. Now, when you get to the lower layer, in the dermis, there are several tactile cells, and we'll talk about it next week. But as far as the epidermis is concerned, Merkel cells are the only tactile cells. And there are also these epidermal dendritic cells, which are immune cells, and they will play a role in phagocytosing pathogens, right? And we can appreciate why you need these immune cells, because if there's anything which gets past the upper layers of the skin, you want to have protection against it. And finally, this stratum basale, it will form what is known as these epidermal ridges. When you think about the term ridge, it's kind of like a raised part on the surface of the earth, you know. So similarly, it's got this pattern called the epidermal ridge that is going to interlock with the underlying dermis in a very unique pattern. So the epidermal ridges will interlock with the dermis in a very unique pattern, and that gives rise to fingerprints, right? So exactly why you have unique patterns, I'll mention it when we talk about the dermis and fingerprints next week, right? But this is going to play a role in the basis for fingerprints. Okay, so with that, we can move on to the next superficial layer, which is stratum spinosum, and this is also known as the prickly layer, right? Because there's something spiny or prickly about it. And this is made up of around 8 to 10 layers of keratinocytes, and the scientists call this the prickly layer is because they found that these keratinocytes had these spiny projections from it, right? They had these spiny projections from it, and these spiny projections were found to be artifacts which were left behind after the processing of the tissue for making these histological sections. So when they were making the skin slides to view under the microscope, there were some artifacts left behind, right? And let me explain what is meant by an artifact. So when they make these slides, the cells will get dehydrated, right? So as they get dehydrated, the water inside them will be lost, but the cells are going to still remain bound to each other, right? And what is the name of that structure which holds cells together? We had an entire lecture on that. What holds cells together? Cell junctions. Thank you very much, right? So my question to you is, what type of cell junction do you think these were observed under the microscope, and they call them as a stratum spinosum? So you notice here is an electron micrographic picture of the keratinocytes, and here you have all of these spiny projections. Even in the light microscope, you see all of these spiny projections surrounding the keratinocytes. So the question for you is, what type of cell junctions do you think are holding the keratinocytes? Are they tight junctions? Are they gap junctions? Are they desmosomes, hemidesmosomes, or focal adhesions? Here's a chance for you to incorporate what you learned in the previous lectures in answering this question. You can turn to your neighbors, TAs, or your notes and answer this. Okay, we have a lot of responses. Let's Kamalani, do you have anyone in your group who has a response for us? Kamalani? Desmosomes, all right. Well, okay, cool. Most of you did say desmosomes, and some of you picked gap junctions, tight junctions. All right. So the correct answer is desmosomes because if you notice this answer slide here, I've mentioned that parts of the body which are going to experience greater mechanical stress, like the heart muscles or the skin or the urinary bladder, they need extra support between the cells, and so desmosomes would be found there, right? So the correct answer is desmosomes. For those of you who picked hemidesmosomes or focal adhesions, you will find hemidesmosomes and focal adhesions in the skin as well, but it will connect cells in stratum basale to the extracellular matrix, right? So that's why you will find hemidesmosomes and focal adhesions, but this was seen in the more superficial layer, and the keratinocytes are linked one to another by desmosomes. Is that clear, folks? Yes? All right. So now let's move on and talk about the stratum spinosum. We see that in addition to keratinocytes, we'll also find that there are these epidermal dendritic cells, which are again immune cells, and they will be quite abundant in this layer. The keratinocytes also will contain melanin granules inside them, and how exactly the melanin pigments get into the keratinocytes is something we'll consider later today, or we may get to it first thing next week, right? So we now move on to the next superficial layer. This is called stratum granulosum. It's made up of around 3 to 5 rows of flattened cells, and this is relatively thin, right? And why exactly it's relatively thin, I'll tell you in a minute, but I want to start by telling you why it's called stratum granulosum. So it's referring to the fact that these cells have keratohyalin granules in them. So these keratohyalin granules, they are going to have a number of functions, and we'll get to it in a second, but let me tell you why the cells in this layer, they are starting to die, and the organelles in them will start disintegrating. So I told you that a feature of epithelial cells is that they don't have any blood vessels. So the only way that these cells get nutrients is that from the dermis, the blood vessels will have nutrients in them, and the nutrients will diffuse to the superficial layers. So as far as the cells are in stratum basale or stratum spinosum, the nutrients are able to easily reach these cells and they can survive. But as they get further and further away from the source of nutrition, not as much as nutrients are diffusing to them, and they will start dying. Now we also will see now the various functions of these keratohyalin granules. There are 4 functions I'll point out for keratohyalin granules. 3 I'll mention in this slide, and the last one I'll mention in the next slide, because the 4th function has to do with the next superficial layer, right? But 3 of those functions I'll mention here. Firstly, it leads to cell dehydration, which is why we see that the cells are getting flattened as they get more and more dehydrated. And why are they getting dehydrated? Because of the keratohyalin granules. There's also cross-linking between the keratin filaments. So in this specific layer, we see that the keratin filaments, which make up the intermediate filaments, they will start getting cross-linked with one another. And finally, the process of keratinization takes place. So keratinization is referring to this formation of this cornified or hardened envelope around the keratinocytes. The reason I have this asterisk here is that this is a very complicated process with several proteins playing a role in this, and I just want you to know that it's complicated, right? But we won't get into the details of how exactly keratinization takes place. So that's what goes on in stratum granulosum. The cells are still alive, but they are starting to die because they aren't getting as much nutrients. And the next superficial layer is stratum lucidum, which means clear layer, so it's transparent. And by the time the cells reach this layer, they're all going to be dead. So you have around 2 to 3 layers of dead keratinocytes. And the reason why the cells in this layer appear clear or transparent is because of L11, which is a transformation product of keratohyalin. Right? So this is the fourth function of the keratohyalin granules, and that is to produce L11, which will lead to this transparent appearance of stratum lucidum. And because it is lucid or transparent, that's why this layer is called stratum lucidum. And this is found only in thick skin, so it's found in the palm of your hands or the soles of your feet. And this extra layer will offer mechanical support against physical abrasion, right? And so when you think about the difference in thickness between the thick and the thin skin, it's like considering saran wrap versus paper towel, right? So that's all the differences. It may not seem like much, but it's sufficient to get the job done in the thick skin. All right, so with that, we move on to the final layer. This is the most superficial layer, stratum corneum, also known as the horny layer. And every time I get to this slide, people start smiling, right? Never fails, semester after semester, right? So this has got around 20 to 30 layers of dead keratinocytes, and they have this scale-like appearance. And I'll show you what this looks like in the upcoming slides, right? This an electron micrograph of the stratum corneum. And this is going to account for 3/4 of the epidermal thickness. So if you see this picture here, it's actually a misrepresentation because it looks like it's making up maybe a fifth or a fourth of the epidermis, but in actuality it's actually making up 70% of the thickness of the epidermis. And this layer will have glycoproteins which are released, right? And that will make it water resistant. And these dead keratinocytes can also secrete certain proteins called defensins, and defensins will play a role in immune function because they are anti-microbial. And I need to go back to this point about why there are 20 to 30 layers. So you may be wondering, why is there a range? It really depends on which part of the body you're looking at, and also the amount of mechanical stress on that area. So for example, let's say you play the guitar, or you're maybe weightlifting, right? In such cases, the parts of your hand which are calloused will have more layers compared to other parts of your body. Finally, this layer also plays a role in offering protection in 3 different ways. It offers physical protection because you have so many layers of cells. It offers biological protection because of these antimicrobial compounds which are released like defensins. And finally, it also offers chemical protection because of the melanin pigments which can absorb the UVB rays and prevents damage happening to the DNA in the underlying layers of the cells. Right? So this is the most superficial layer, and so we have started at stratum basale and we have worked all our way up to stratum corneum. And so cells in stratum basale, as they differentiate into keratinocytes, they'll be gradually moving upward to stratum corneum. So here's a think, pair, share activity for all of you to consider. How many of you have heard about henna, dye, or paint? Yes? All right, few of you here. Previous class we had more people who were aware of this. So this is referring to the dye made from plants, right? So it's popular in Asian countries, and it's not only used to do skin art like this, but it can also be used historically to dye fabrics and things like that. And to get on something this intricate, it might take, I don't know, a couple of hours, depending on how skilled you are. But this henna dye will just wear off in a couple of weeks, right? So the question is, why do you think it doesn't last longer? Why does the dye come off in a couple of weeks? You can turn to your neighbors, TAs, or yes, Oh yeah, you turn to these two people and ask them what the answer is. All right, folks, let's bring it back together. Okay, Chloe, anyone in your group has a response for us, Chloe?
It's because the skin sheds layers of sebum.
That's very good. So the answer said was the skin is shedding the most superficial layers, and that is correct. So it takes around 7 to 10 days for the cells to move all the way down from stratum basale up to stratum corneum. Right? It takes 7 to 10 days, and once the keratinocytes reach the stratum corneum, they're going to stay around there for around 2 weeks before they are eventually shed or they are washed away. Right? So it takes 2 weeks for stratum corneum to be replaced. And so in a period of around 45 days or so, your entire epidermis is replaced. And so that's why if you get a tan over the summer, it doesn't last too long, right? It's gone within a month or two. And so what you're seeing here is a picture of the electron micrograph of the epidermis, and this looks like shingles on a roof, right? That's what it looks like. And because our body is shedding skin regularly, in a lifetime we'll shed around 40 pounds of skin, right? So that's the amount of skin we shed in a lifetime. So now we're going to move on and talk about what are the cells found in the epidermis. I already told you that keratinocytes, they are the most abundant types of cells in the epidermis, but I want to spend a few minutes talking about the other cells we encounter as well. You also have these cells known as Merkel cells. These are going to be found in stratum basale, in the deepest layer of the epidermis, and you don't see Merkel cells pointed out here, but I did have it labeled in the previous picture. So Merkel cells are only one type of touch receptors, and they are the only touch receptors seen in the epidermis, right? The other receptors, we'll talk about it when we get to the dermis. And they will interact with these free nerve endings, which are capable of detecting senses like pain. You also find that there are these immune cells known as the Langerhans cells or the dendritic cells or epidermal macrophages. These are all cells which can attack pathogens if any pathogen were to get past the superficial layers of the epidermis. And lastly, I want to point out the presence of melanocytes. These are the cells which produce the melanin pigment, which gives us the skin color. So where you find melanocytes is going to be in stratum basale. So this is in the deepest layer of the epidermis, and I told you it's going to make up 10 to 25% of the cells in stratum basale. And when these cells, they are exposed to UVB rays from the sun, it has to be the UVB rays because you have UVA and UVB. So in response to the UV rays, UVB rays will produce the melanin pigment. And these melanin is going to accumulate in vesicles called melanosomes, and it's going to be transported in these melanosomes. And from the melanocyte, these vesicles will then enter into the keratinocytes, right? So how exactly these melanocytes enter into these keratinocytes, I want to highlight that by showing you a short video clip.
In this scene, a melanocyte develops two long dendrites, which introduce packages of pigment granules, or melanosomes, contained within vacuoles, into a flat keratinocyte. While the individual melanosomes are too small to be seen in the light microscope, the transferred vacuoles, containing many melanosomes, are visible. The melanosomes are left behind as the dendritic processes start to withdraw. The vacuoles open up inside the keratinocyte, dispersing pigment into the cytoplasm. Let's watch the pigment donation again.
Notice how the pigments move into the keratinocyte. Melanocyte is up there and the keratinocyte is down there. All right, so the question for all of you is, how are these melanosomes transported towards the periphery of the melanocytes? Are they transported by which type of motor protein, using which cytoskeletal protein, right? So that's the question we'll be answering when we come back next week. All right, let's stop class for now, and I hope you all have a great weekend. Thank you all for your time.
So I'm submitting an application for an internship.